Hello to one and all. Here today for the 10th episode of Immunology for Everyone podcast by SKA. The journey has solely been to deliver immunology concepts and facts like never before. Your host and epitome of dedication and passion towards the field has been motivated to share his experiences, knowledge, and influences for those who have a fire similar to his. Let us venture ahead with him and understand how our immune system generates and modulates various cells and their functions to help us fight foreign invasions. Don't forget to invite your peers, share with your fellows, and keep sending in your queries. Here is SKA on his note again.
Hello, everyone. Welcome back to the podcast Immunology for Everyone. So, dear friends, we started discussing that the antigen specific immune responses against any invading pathogen are driven by the antigen specific T cells and B cells. So, we have already discussed the T cell activation and generation of cell mediated immune responses. Now, when we start talking about B cells, we must understand that these cells are responsible for driving the other arm of our immune response, which is called the humoral immune response. This implies that the B cells perform their protective function via the effector molecules, which are called the antibodies, both in the form of a surface immunoglobulin, which is the surface receptor on B cells. And as a surface immunoglobulin, this molecule helps the B cell to bind to the pathogen directly and make the B cell as one of the professional antigen presenting cells. And secondly, the soluble form of antibody, which is the secreted antibodies, which circulate in the blood, providing protection against the pathogen against which they have been generated. Because once generated, these antibodies keep circulating in the blood for quite long time, sometimes even for months, and even after the clearance of the pathogen. So the binding capacity of this antibody to antigen allows it to perform its protective function in many ways. For example, the antibody can directly bind to the protein or the glycoprotein on the envelope of viruses and neutralize the virus, which means that on binding to the virus, it decreases the replication capacity of the virus by blocking the attachment of virus to the host cell and also preventing its penetration into the cell membrane. Or in the other way is that it can interfere with the uncoating of the viral envelope. Like whenever the virus enters into a cell, first it has to uncoat itself. The nuclear material is released inside the cell, and sometimes this nuclear material, which can be DNA or RNA of the virus, can get integrated into the host genome, like in HIV, or it may remain extra chromosomal, like in case of HCV, or maybe this SARS-CoV-2. And still, the uncoating is very important for start of replication of the virus. So then we also know that it mediates the antibody also assist in facilitated phagocytosis of the pathogen, because once it binds to the surface of the pathogens, then it in a way a process of opsonization, which is taken up by the phagocytic cells and degraded. And then this antibodies are also known to help in antibody mediated cell cytotoxicity, which is also known as ADCC, which is a major activity of NK cells, how they kill the target cells. So we will talk about this ADCC sometime when we talk about the NK cells. So beside all these protective mechanisms, the circulating antibodies also help in specific diagnosis of the infection, because they have the antigen specificity. And we all know that there are many very sensitive techniques which are called the serological assays, by which we can detect the antibodies circulating in the serum of the individuals who might get infection. And there are many examples, like we look at the anti-HIV antibodies in the serum, anti-HBV, HCV, and many other pathogens so far it's possible. So the detection of antibody by some sensitive techniques like enzyme linked immunosorbent assay, which is a very sensitive and specific technique, also called ELISA. And nowadays we have many rapid assays by which we can detect the antibody, and this helps in diagnosis and start off in the treatment by the physician. So similarly, uh, there are many other functions of these antibodies. So let's understand what are these antibodies. In terms of antigen specificity, the secreted antibody is identical to the B cell receptor of the cell that ultimately secretes it. So B cell get activated and then differentiates into antibody secreting plasma cells, and these antibodies which are produced, they have the same antigenic specificity as it was in the B cell receptor, which was found initially on the mature B cell when it comes out of the bone marrow. But there are few structural differences in the surface bound immunoglobulin molecule and the secreted antibody molecule. In the case of surface immunoglobulin molecule, one end of the immunoglobulin molecule is a hydrophobic membrane anchoring sequence, whereas uh, when it is secreted antibody, that end has a hydrophilic sequence, and that allows its secretion. uh, if we look at the structure of immunoglobulin molecule, the antibody is typically a Y-shaped molecule with a unique chemistry, and this unique structure allows the antibody to carry out its major functions. One of the tasks which it performs is that it binds to the specific antigen, and we know that it binds to the pathogen or any antigen or the surface glycoproteins of the viruses and all by the antigen binding domains of the antibody. And the second major task which these antibodies they do is they can anchor to a limited number of effector cells through its stem. And we'll talk about the details of the structure. And the stem of the antibody, which doesn't bind to the antigen, but it helps the immunoglobulin molecule to anchor onto certain effector cells, and in that way it also helps to activate and send out the activation signals. So in this context, please understand that each of these tasks is carried out by separable parts of the immunoglobulin molecule, because as the antigen binding sites, they lie on the N terminal of the polypeptide chains which constitute the immunoglobulin molecule. So there are two arms and a stem of the Y molecule which are called the variable regions. So the antigen binding portion lies in the variable region of the antibody molecule, whereas the stem is not that variable. So it basically is more constant. So it's also called the excuse me, constant region of the immunoglobulin molecule. So this part doesn't take part in the antigen binding, yet it mediates the binding of the immunoglobulin molecule with some effector cells like NK cells, macrophages, and and it uses specific receptors which are present on these cells, which are called the FC receptors, and it that way it passes on some kind of activation signals to these cells over which it anchors. So as we see that the chemistry of immunoglobulin molecule is very important for its functions. So let's discuss the chemical structure in a bit more details. So each immunoglobulin molecule is made up of four polypeptide subunits. Each subunit has two identical long chains which are called the heavy chains, and two identical smaller chains which are called the light chains. These chains are held together with disulfide bonds as well as some non-covalent bonds which form certain conformational domains in the polypeptide chains. So the amino terminal, as I said, has a domain which is called the variable domain. So that is with the heavy chain as well as light chain. They both of them they comprise a antigen binding domain together. And so there are two identical antigen binding domains in an antibody molecule, which commonly form the fragment antigen binding or the Fab domains. And the variable region further contains certain hyper variable sequences in them, which are called the complementarity determining regions or the CDRs. So there are three CDRs present on the heavy chain variable domain, three CDRs present on the light chain, and the six ones they act like claw and they hold the antigen. So the specificity, antigen binding specificity, actually lies in the CDRs, and they take part in the antigen binding. On the other hand, the carboxy terminal of the heavy chains, which are the long chains, they form the FC domain at the end, which is the carboxy terminal, and that particular fragment is called fragment crystallizable. Because it's a constant region, it can be when the enzymes cut the antibody, the lower portion can be crystallized, so it is called the fragment crystallizable or the FC domain. And this one actually binds to the FC receptor, which are present expressed on some of the effector cells. Excuse me. Sorry for my bad throat. And then the peptide sequence, which is between the Fab region and the FC region, that is in the heavy chain, that is called the hinge region. So the unique structure and position of hinge region provides segmental flexibility to the two arms of the antigen binding arms of the immunoglobulin molecule. For example, they help in cross linking of the two antigens or binding of two antigenic determinants which may be present apart on a single antigen molecule which may be big. So it gives a flexibility to the these arms. So, if we go further and try to understand the functioning of these antibody molecules, it has been shown that depending on the chemical nature of the heavy chains in an antibody molecule, there are five primary classes of immunoglobulins, and these are the IgG, IgM, IgA, IgD, and IgE. For example, the IgG molecule has a heavy chain that is known as the gamma chain. IgM has the mu chain. IgA has the alpha chains. IgE has the epsilon chain, and the IgD has the delta chains. So while we have the five different types of heavy chains, and that makes five different types of the immunoglobulins. There are only two types of light chains, either kappa type or lambda type. So in a immunoglobulin molecule, you can have any of the five heavy chains, but any one of the two light chains, either kappa or lambda. So, as we know that there are two binding sites in each antibody molecule, the antibody classes they differ in the valency as a result of different numbers of these monomers which are joined with the J chain to form a complete protein. So, for example, the in humans, each functioning IgM antibody has got uh, five monomers attached with the J chain. So it's a pentameric antibody. And then, being a pentameric, it has 10 binding sites. So the valency of a IgM molecule is 10. Whereas IgA, you have two monomers, they are joined together with a J chain, and that way it has 4 valency. Whereas the other immunoglobulins like IgG, IgD, and IgE, they are all monomeric, so they have a valency of 2 only. So they can bind uh, two antigens at the same time. Another interesting aspect of antibodies is that they can switch their class during an ongoing immune response. And this phenomena is called the class switching. So, class switching basically occurs after the activation of a mature B cell via its membrane bound antibody molecule to generate the different classes of antibody, and all the classes will have same variable domains as the original antibody which is generated in the immature B cells during the process of development. So they possess distinct constant domains in their heavy chains as the classes are different, but the variable domain remains the same. So they will bind to the same antigen even if they change their class. So as we see that the naive B cells, they display mainly the IgD as the surface immunoglobulin, but once they are activated, the primary response is always the secretion of IgM antibody, and then as a subsequent exposure, and there becomes a antibody class switching, and the IgM expression is down regulated, and IgG or IgA or IgE type antibodies are expressed. So this is a class switching. uh, for example, in a viral infection, uh, if you are trying to detect the antibody class, what kind of antibodies are generated. Sometimes, because I told you that the antibody keeps circulating in the system even after the pathogen is cleared. So viruses normally get cleared very quickly the after the acute infection, but the antibodies remain circulating. So in case you want to see distinguish between a very recent infection or some past exposure, then the lab will do kind of IgM and IgG, and if only IgM is detectable, then you may say that it's a current infection, very recent infection. Whereas if you see IgG only, then it can be a past infection, past exposure. So the virus has already been cleared, but the IgG remains. So that's a very important information we get by looking at the classes of the antibody sometimes, which helps us understand whether it's a recent infection or some past exposure. And it is very interesting to know that during class switching, only the constant region of the immunoglobulin heavy chain changes, while the variable regions remain the same. And therefore, the antigen specificity also remains the same. So, this allows different daughter cells or the progeny of the activated B cell um, uh, to produce antibodies of different isotypes and subtypes with the same antigenic specificity. Now comes, once the B cell is activated, the plasma cells, they differentiate into the plasma cells, which are the antibody secreting plasma cells. So, besides class switching, the B cell activation start presenting the antigen on their surface, which leads to their crosstalk with the antigen specific T cells, which are the helper T cells, as well as the T follicular helper cells which are present in the follicle. So there is a follicle formation. The antibody forming plasma cells undergo maturation with repeated interaction with the T follicular helper cells, leading to generation of a long-lived plasma cells, which keeps secreting antibodies for a long period of time. And if you want to see how long the antibodies can sustain in an individual which has been exposed, then it all depends on whether there is generation of these long-lived plasma cells is there or not. And some of the activated B cells, they basically become the memory B cells. So they go like into a dormant state, although they have surface immunoglobulin, which is specific for that receptor, and they do not make any antibodies, but they remain, and they are the long lived memory cells, and they help in quick response whenever there is a subsequent exposure with the same antigen. So at that time, the memory cells, they start responding very quickly to the antigen, same antigen or same pathogen, because they have the memory of the previous exposure. So that is also basis of the vaccination, and we'll talk about all these importance of generation of memories, B cells, and protective response by the vaccines sometime in the later episode. So, for the time being, let us understand that the plasma cells, they undergo further maturation, which is known as the affinity maturation. And this process involves a phenomena which is called the somatic hypermutation, and that basically is facilitated by the interaction of the B cells with the T follicular helper cells. We'll understand the process of affinity maturation and somatic hypermutation in more details in the next episode. So today, let's close here. But what is the carry home message? One thing we discussed was that the B cells are the ones which basically differentiate into the plasma cells, which produce antibodies of the specificity of their B cell receptor initially. A B cell has got a immunoglobulin receptor, it has some antigenic specificity. So after maturation, after the differentiation into antibody secreting plasma cells, they produce the antibody of the same antigenic specificity. The second thing we understand is that the secreted antibodies are the functional molecules which perform various effector functions like removal of the pathogen or neutralization of viruses and many other functions as we discussed. Then we also understood that there are five different isotypes of the antibodies, which are the IgM, IgG, IgD, IgA, and IgE, and they basically, these isotypes are depending on the chemical nature of heavy chains, and they can switch from one type to another during the ongoing uh, immune response. And then we understood that each isotype of antibody have at least one unit of immunoglobulin molecule. Okay, as we know that the IgM has five monomers, IgA has two, whereas IgG, IgE, and IgD, they all have one monomer, and all these immunoglobulin molecule structure, chemical nature helps to perform its important effector functions. Then the last thing we talked about was that the activated B cells, they basically differentiate into plasma cells, which are the antibody secreting cells. These plasma cells, they further undergo affinity maturation and clonal selection. And next time, what we do is we'll discuss the affinity maturation of these activated plasma cells. So till then, you must go and read some of the good chapters in your immunology books. And next time, come prepared when we discuss very interesting aspect of affinity maturation, hyper mutation, which leads to generation of those cells which are secreting very high affinity antibodies, which have a better capacity to neutralize and clear the pathogens. Till then, thank you very much. Thank you for listening, and goodbye.